Yes. I should shut up and let you say hi, Juliet.
Hi. Great to see people are actually signing in, and we're excited to have you. I'm really happy that Angie has been, you know, promoting so much content and it scientifically accurate content. So, and I'm happy to be a part of it. So, I'm excited.
Well, as you know, Juliet, I just can't get enough talking about virology with people that I've known for 25 years because of virology. Vincent and I, our PhD mentor, Juliet and I's PhD mentor, filmed a thing last night that will be coming out probably next week, I think. But that's basically all of my shows. I just get together with people and I talk about viruses in different ways. And Juliet and I, Dr. Morrison and I should introduce Juliet and myself. I am Dr. Angela Rasmussen. I am in home casual mode now, which is why I'm wearing a Seahawks t-shirt because I'm from Seattle originally, and the Seahawks are going to the fucking Super Bowl this weekend. Juliet
Are
Morrison
you
is my
serious?
good.
I
Yes.
did not
Of
know
course you
this.
didn't know that.
Of course, no, but I mean, I have a special place in my heart for the Seahawks because
I
I
know
I
you do.
watched, and I have that Marshawn Lynch jersey, and I love him. So yeah,
That you wore
wow,
to my wedding.
yes,
Um,
good job. Go
yeah.
Seahawks.
No, I actually thought I considered tonight wearing that Marshawn Lynch jersey for this talk, but to be honest, like my ancient like Seahawks shirt is a lot more comfortable than like jerseys. So I left Marshawn in the closet with Richard Sherman and Bobby Wagner and the rest of them. But yes, so the Seahawks, Juliet and I throughout our career, we went to graduate school together in New York. And then later, after our postdocs, we worked together at the University of Washington together. And that's when Juliet lived in Seattle. And I lived in Seattle. And while we were there, the Seahawks went to the Super Bowl. And then the next year, the Seahawks lost the Super Bowl to the Patriots. Boy, was that a disappointing Super Bowl. So now it's the rematch. Juliet, are you still there? Your camera went off.
I know. I'm trying to turn off my notifications. I just realized that like things keep popping in. I apologize. I'm just trying to
Oh, no worries.
mute WhatsApp. Um, all right, what do
No
I
worries
do
at all.
here?
I just wanted to make sure last night my little show was plagued by internet problems on my end. So
Mhm.
I just wanted to make sure like I didn't need to start just chatting to keep people busy until the real show comes back.
Yeah, okay. Yep, all right. I have muted um WhatsApp. Okay, great. Yes.
Yeah. So anyways, back to introducing you. This is Juliet Morrison, Dr. Juliet Morrison, Professor Juliet Morrison from the University of California, Riverside. We went to graduate school together. Then we worked at UW together. Between that, Juliet went off and did her own postdoc at Mount Sinai with Adolfo Garcia Sastre, who's one of the world's greatest living influenza virologists. She studies now actually a lot of different viruses, but she's a macrophage kind of girl. She's like me, she's into the host. And she's just picked her own cell type that she likes. We're collaborators and we've been friends for a really long time. And that's just kind of what we wanted to do. We feel like right now, I feel anyways, but I think you agree with me, Juliet, that, now we just really need clear, effective, accurate science communication from experts. And, doing that at a scientific conference or reading even a lot of scientific communication for the layperson, it's pretty boring, and sometimes it's boring for the non-layperson as well. Um, so I thought one thing, and people have been telling me that it's actually quite helpful to just hear people kind of talk to each other like normal people. So we thought we would have a show where we just pick a virus topic, and we'd make thematic cocktails, and we'd get together and we'd just chat like a couple of experts who are also old friends. And I think funny and interesting people. Um, So today's topic is measles, which is certainly in the news. And right before I went live, Juliet and I were just discussing the fact that neither of us know very much about paramyxoviruses. And measles is, in fact, a paramyxovirus. It's a negative sense, single stranded RNA
Granted.
virus, which means that it's one long piece of RNA that is the opposite sense from what is coded. So it has an interesting replication cycle. And it's basically like an influenza virus,
But
except
simpler.
instead of having 8 segments of genes, it's just all stuck together in one. And that's kind of the extent of my knowledge of paramyxoviruses. At one point, I did learn a little bit more about how to clone them for a consulting exercise that was like, could you make a bioweapon? In theory, I could. I figured out how to clone a paramyxovirus. You'd have to actually have a paramyxovirus that could be a bioweapon, which I think is more of a challenge. But the cloning part, I got the theoretical part down. But as you know, Juliet, sometimes the execution of that particular lab procedure, did not come as naturally to me as other things like bullshitting on Substack.
I mean, it happens, right? I mean, we all have our techniques that we are good at and or not good at. Like I hate microscopy with a passion. Like I just find it so tedious. But I love me some Western blots and I love to clone. Like those are like my favorite, and plaque assays. Plaque assays, zen feeling when
I
you
like
do
the
that.
I love the plaque assays. That's why
Yeah.
I love working in containment because it is like the most peaceful thing that I could possibly be doing. It's just like me and a plaque assay. Like, can you imagine if I had said something like this in grad school that I would think that it would be meditative and comforting to do like 8 gazillion serial dilutions? But it kind of
No,
is. It kind of is. Like I did so
it
many of them in
is.
grad school. I was like, oh my God, my hand's gonna fall off. But I get carpal tunnel syndrome for life. But now that I'm a PI and I don't work in the lab as much anymore, when I do it, I mean, I'm always offering like, "Do you guys need, like, me to help you titrate your growth curve?" Like, it's like, "No, we don't really want you in there with us. Like, you're annoying."
Yeah, like, at some point, you become the weakest link, when it
Oh,
comes
I
to
I don't know where
experiments, which is
anything is.
embarrassing because, like, I really am not. Like, I'm like, "Guys, I will come in here and show you that you can do 30 plaque assays over the course of the day." And, yeah.
My lab, like, they offer to do things, I think, in the lab. If I'm gonna go do something in the containment lab, they're like, "Do you need me to split those cells for you?" And I'm like, "I mean, I could do it." And they're like, "No, no, we can do it for you. One of us will be available." I'm like, "Y yeah, this is actually the best part of being a PI. I don't have to split the cells at all. I can just go, like, do the experiment, tell other people how to do the experiment." Well, sometimes I show people. And
Yeah.
now I'm like, "Oh, I'll come in and help you." And they're like, "You can come in on your own time."
Yeah,
Like,
I think it's not even that they I mean, because they know that we are actually the pros at this and we know what we're doing. I think the issue is like who wants to be supervised by their boss like right there? Like I mean, like it's a lot of work. Then they feel uncomfortable. So I guess it. Unless I'm training people, I try to I'm not I hate being micromanaged, so I don't micromanage people. But like I've realized that yeah, my trainees, the best way for them to learn is to actually kind of like fuck up and then realize like, that's what happens when I don't follow the protocol. Yeah, that's what happens. But I don't need to be the person who tells you. Like I think you need to learn it and then you keep going and take ownership of your own stuff, you know.
I agree. I mean, that's how we were trained, and,
Yes, exactly.
I have to say I also apply the rack and yellow method in my lab, which is, I mean, I so for everybody watching, my graduate school, my PhD work project, was a resounding non-success for quite a long time. And it's not that I wasn't good in the lab or I wasn't competent. It's just that my experimental system didn't work.
It didn't
And it
work.
didn't work in a way that required me to do 2 years of experiments to figure out why it didn't work. It was a transgenic mouse that receptor transgenic mouse in virology is a mouse or an organism that expresses the receptor for the virus on the surface of the cell. So that makes the virus able to enter the cell. It can cause an infection. There's no mouse model that's really good for the common cold. So you can't study asthma and cystic fibrosis and stuff in it. So that's what I was trying to do. And I needed a transgenic mouse to do it. And it turned out that my transgene was being expressed on the inside of the cell, not the outside. So no transgene to attach to, no receptor to get into the cell. My mice weren't getting infected. And it took me 2 years to figure out that that was what was going on.
But I will
So,
give it to you that, like, Angie, that actually, I I feel like a lot of people would not have come up with that. And I think it was because you had, like, the understanding of, like, flow cytometry that, like, at that point, I feel in virology, people weren't really using flow as much in detail. Like, it was like the flow cytometry and the microscopy, from what I can remember, that indicated that this thing was not being expressed where it was supposed to be, and it was not on the cell surface. So it could never act as a receptor for rhinovirus, and so the whole situation, everything that you were doing, just wasn't going to work. But
no.
I don't think the average PhD student would have picked that up. Um,
In
yeah,
fairness, I was sort of a mid-career PhD student at that point. It took me a couple years to figure it out. So I was 3 years in when
yeah.
I finally got that. It wasn't even, oh god, I just love old virology stuff. It wasn't just the flow cytometry data. So what Juliet's referring to is the fact that I basically did everything I could think of to figure out why these mice weren't getting infected, because I knew that they had the transgene. I knew that they were expressing it at the protein level, at the RNA level. I did a goddamn southern blot. I did a northern blot. I did a western blot. I blotted around the compass to look at protein, at DNA, at everything. I did PCR. I was doing all kinds of stuff to prove that the transgene was there and the receptor was being expressed. And it was. And then I did histopathology. I cut tracheal sections of the mice and I stained them, and boy, it was beautiful. It was one of the most beautiful stains I've ever done in my life. You could see the receptor, which is called ICAM1, just lighting up the tracheal epithelial cells in the, facing the lumen of the trachea, but all through the cells. That's not abnormal for receptors, because you will see the receptor inside the cell if you stain it,
Yes.
because that's where the receptor gets made. So it gets made and then moved to the surface of the cell. And so you would be able to see it in both. And I did see it kind of by both, but you can't really tell if it's sticking out of the membrane. Like, my microscope wasn't that good. And like Juliet, I too hate microscopy. So I was like, I don't see what the problem is. Like, why the fuck is this thing not getting infected? And like, not infected at all. I just was infecting these mice and there would be no virus at all growing in them. And this was a mouse adapted virus. Like, there was
In
no
a
reason it
sense,
couldn't
it
grow.
is almost seems like they were resistant, and as opposed to susceptible, which is like a theme that I love to talk
Because
about
it
in
turns
like
out they were.
yes.
They still weren't expressing the fucking receptor. Like,
They
it
weren't.
sucked.
They
And
did
it
not.
took a
You
long
need to
time.
have a receptor on the surface for a virus to be able to enter a cell. Like, if it cannot bind to like a virus is a key and the receptor is a lock, it cannot get into the cell unless there is something on the surface of that cell that is like a beacon to that virus. So yeah, your thing was expressing really well, but it was just not expressing on the surface of the cells. So unfortunately,
Yeah.
um,
Yeah. And it took a
but
long time.
it took
So
a
I
lot. It
found
took
that out by doing this. I was desperate. I ran out of things and I was like, I can't explain this. And I was a graduate student. So, of course, I knew that my committee would expect me to explain it. And I just, before I went to graduate school, I worked as a technician at an immunology company. So I did a bunch of flow cytometry. And a flow cytometer is a device that basically takes cells and it puts them through a tiny little aperture, a tiny little microfluidic system that can look at a single cell at a time. And it blasts them with lasers And if you've got fluorescent stuff in there, where the fluorescence detects, can tell you what kind of cells they are effectively. So I knew how to use a flow cytometer. There was one at Columbia. I was like, you know what? This is how we look at T cell receptors. This is how we look at T cell co-stimulatory factors. Those are proteins on a cell surface. Let me try to dissociate these cells from the mice and stain them with antibodies and then run them through a flow cytometer. So I did. And guess what? No receptor. So to confirm that and to confirm that they were completely not expressing any functional receptor, I then cultured these cells. And hats off to old school virology, did DEAE dextran transfection of in vitro synthesized T7 in vitro synthesized, mouse adapted rhinovirus RNA and transfected that into the cell using this old timey method. This was Vincent's, sort of new and improved family recipe. And by new and improved, I mean like in maybe 1987 or something. Um, but anyways, it's a family recipe for making picornaviruses. And so I transfect these cells. I put the genome of the virus into those cells. If it uh, if it will be made when it gets into those cells, the viruses will be made. It's essentially a reverse genetic system. Um, New virus will then go infect other virus or other cells in the culture dish around it. In HeLa cells, which are very susceptible to rhinovirus, tons and tons of rhinovirus. In my cells, very little, below the limit of detection of the plaque assay. Um, there was like 1 or 2 plaques. So input virus, basically. It wasn't spreading. It wasn't the cell's problem. Virus could grow in them, but it couldn't spread in the culture because no receptor. So that's an example of a functional experiment that was actually a great experiment now that I think about it. I mean, that was a great way to validate the flow cytometry data and really show like the hypothesis that it's not being expressed on the surface was correct. Like that was a great experiment to test that hypothesis. But it generated what we call negative data, and my system didn't work. And that was the point where we decided to pull the plug and start all over. And then I had to mouse adapt a new rhinovirus from scratch. So I was upset about this, and I was upset about spending 2 years of my young mid-20s life, uh, you know, dealing with this whole thing only to find out that my project was hopelessly fucked. And you know what Vincent told me? He was like, "It builds character."
Oh God.
And
Please.
I mean, I remember at the time I was like,
I
"Builds
can't
character? I
remember
can't
that. Like when
fucking
you were
publish
so
shit. Like,
mad.
what
You
the
were
fuck?"
so mad.
But you know what? It did.
It
It
does
taught
build
me
character.
it
It
taught me how to
does.
be a really good scientist. I
Yeah.
followed that I followed that trail to the end until I solved that problem entirely experimentally.
Yeah.
And that's really like what a scientist should be. I'm not
It
to say
is
that I'm like the
exactly
poster girl for that, but that's the training. And I
that.
think that,
That is a
going
training.
back
And I
to the
feel like
original
a
thing, I
lot
think
of,
unless you screw up,
yeah, you have to screw up to actually appreciate the knowledge that you're gaining from screwing up, and to actually like sort of validate the fact that you have learned something and learned enough to actually troubleshoot. Like if you can't do that, then you are not actually a scientist. Like you are a robot. And so like for me, like it's the independence that tells me, like I know that my students are doing a good job when they are the ones who are like, I thought about this thing, even if at the end of the day it might be a wrong thing, but the fact that they're trying to troubleshoot and they're thinking about it and they're coming up with stuff that is actually pretty plausible, uh, that is very exciting to me. Like that's what science is. It is the figuring out why something is broken, because most things are broken and no experiment ever works. um yeah.
I have to say my students, every single one of them is so disappointed that it turns out real science is not like cloning GFP in your undergraduate molecular
Yeah.
biology lab class. Guess what? It worked for me the first time too when I was 20 in 1990. something. Um,
I don't even know if you should tell people. Like
I'm trying to remember
1841?
what year. Actually, no, it would have been either 1997 or 1998. I think we did the clone GFP into Puck 19 experiment
Mhm.
in the Smith College molecular biology advanced lab class.
And those things are designed to make it work so that you can feel good about it. And then it kind of like sets up people to think that that's what science is. And science is mostly failure. Like I speak with I just had like two meetings like with two of my master's students today. And my master's student was like, "Oh, like maybe this week it will work." And I'm like, you know what you're doing is actually the process. This is the science. Like, I know it doesn't feel like you're moving past this stage yet, but like what you're doing is that you're growing. Like, you have learned now all this stuff about DNA extraction, all about the things that you weren't really paying attention to before, and now you understand it. So like you have this increased understanding of these techniques that you didn't even understand before so that you can troubleshoot. And so you are actually displaying you're a scientist. Like you are displaying these um traits that tell you the curiosity, the need to figure out why something is broken and how you can address that broken thing. That's like it's amazing. I just wish that people could be rewarded for just like learning and becoming better thinkers, but it doesn't really work that way in our society. But kudos to
It
my
never really
students
works
who, when
that
they step up
way.
and they do that, I'm so proud. I'm like, yes, like, oh, you thought about this before I told you. Like, that's great. Because I will sit there for like a month and just let you flail around because like that's the only way you're going to learn.
Yeah. I mean, I think that, failure is a really important part of learning. And I have to say that for me, a lot of my failure in grad school was responsible for a lot of my success later. And again, I'm not like one of the most successful people in the world, but I think I've learned how to be really good at what I'm good at. And I think I've learned also how to work with people who are good at things that I'm not good at. And I've also learned, I mean, and this is, a good segue into what our actual topic was, because
Oh my God, right.
when we
Yeah.
have the conversation that's like, join us, trainees, to talk about the Rock and Yellow method of letting you fail forward and discover like how horrible science is and then how wonderful it is. And you will have that epiphany, I promise, but you got to walk through the fucking valley of death to get there.
You
Anyways,
have to.
um, let's talk about measles because this is
Okay.
actually quite relevant. It is my
Hmm.
training that has allowed me to push back against a lot of this stuff. Because at some point in my training, and in Vincent's lab and every experience I've had since then, and since Vincent, I've had a lot of really shitty bosses. Um, from
We've shared some
the
of
good
them.
ones
Oh God.
From the good ones, like Vincent, and the really shitty ones, like Michael Cates, our mutual boss, who was stripped of tenure and fired in 2017 from the University of Washington for spending federal grant money on coke and hookers. Um, Basically, that is what both of us have survived. So for me, I think of this all the time, given the situation that our world is in right now. I'm facing people who are far worse than, even some of the worst people that I've had to deal with in academia. They are pretending to be bigger experts than me. But fortunately, I was trained by Vincent Racaniello, who's a bigger expert than all of them and who's extremely good at training people and teaching them how to be virologists. And I think, Vincent saw something in both of us that we both have a lot of aptitude and passion for virology. And I think about this every day because in many ways, I didn't realize it at the time, but I was trained for this. Science isn't
Yes.
about just like thinking all day. As you pointed out, science is about like doing things and like having that curiosity, but having it and then following through on it, making those discoveries, and most importantly of all, using the method to do it. For me, when I look at, like, say, RFK Jr. giving a talk, there is there's the fun Angie part of me that's, like, not the scientist part. That's like, "Look at that motherfucker." Just mocking him and thinking, like, he looks like a piece of beef jerky and etcetera. Like, why is this guy talking about my field of expertise? And then there's the part of me that is not angry necessarily or going to attack him on that, but the part of me that's most offended is the scientist in me that's like, "There's no data on that." And I have to say, I have spent so much time this past year, my new hobby. I mean, some people get into ham radio or like
Ham
Civil War,
radio, okay. Like, how old
Civil
are
War
you? Like,
reenactments. I don't know what are hobbies like knitting.
I,
Oh, Smith College hobbies: knitting, fencing, uh,
that
poetry
sounds
readings.
that sounds very, very women's liberal arts college. Yep. Hmm.
I mean, it sounds very barred as well. We both went to liberal arts colleges. Yours
That's
is yours
true,
is more
but
liberal arts than
mine
mine.
was co-ed.
Crocheting. Look, one of our viewers just mentioned crocheting.
Oh
I
God,
don't know how to
yes,
do
that is
any of
quite the thing.
that.
That is, yeah.
It was big at Smith. There was the fencing club was called Hell's Bells with an E. And there was also a knitting club called the Knitwits. Um,
Oh,
but
oh,
yeah, but like
that's cute.
my hobby is like being awake all night and like obsessively learning everything there is to know about my enemies. And
I know,
it's
you're so weird. I love it.
no, but I'm like, I seriously like I think that this asshole's gonna do this into like hypothesis testing. I'm like, let me test that hypothesis. And then I like spend, all night basically, like looking at papers that would potentially prove this. I literally spent an entire weekend writing a response to RFK Jr. because he responded to me on Twitter by simply posting a fucking paper from like
Wow.
human reproductive biology, which I looked it up, impact factor like 25. And I was like, okay, I don't know anything about human reproductive biology. Like, this is definitely
So
a
this is
big
a
journal in
legitimate
that
journal?
field.
This is a
It's
legitimate
a
journal?
journal. Yeah,
Oh,
I
okay.
can't remember if it's
No, I
widely
mean, that's a very high impact factor. That's impressive.
it's huge
But I mean,
impact factor. It's like nature medicine level.
yeah,
It's
but at the same
crazy.
time, I don't know if our I mean, I'm sure many of our viewers are understand impact factor, but I think others might not. Where that you can actually game the system to get a high impact factor if you incentivize people to write review articles and all this other stuff. You can somehow build a standing that might not necessarily mean that you are the most selective of journals. I don't know anything about this journal though, so I'm
That's
not.
actually correct.
Yeah.
So here's the problem. It's not that he posted a bad paper. Paper seems totally fine. I mean, I'm not an
Okay.
expert in that field, so I can't really judge, but like I'm an editor in chief of a journal, so I do know how to read an academic paper. And no, the journal's no problem.
Okay.
The paper wasn't about what he was posting about, which was vaccines decrease teen sperm count. And I said, well, it's kind of weird that, you
Who
know,
cares? Like
lawyer,
I don't
the
want
the
teens
HHS
breeding.
secretary,
Like,
who's a fucking anti-vax lawyer, is saying that, vaccines he was actually implying, but the vaccines, like, decrease teen sperm count. And I'm thinking to myself, how do you do this experimentally? Well, actually, you get a bunch of adults to tell minor children to jerk off into a cup. And so I brought this up that, I don't think any IRB, an institutional review board, the ethics board that reviews clinical trials that involve human participants, especially, and when they are minor children, they're considered a vulnerable population because they cannot consent legally to be part of this trial. And you're talking about having adults instruct teenagers or children how to jerk off. Um, Unless there's
so
some sort of
creepy.
health reason why you need to do that, I don't think we need to be encouraging teenager I don't think we need to give a fuck about teen sperm counts. We know that vaccines have no effect on adult sperm counts.
Yeah.
So
And I
fine.
think the last time
Kennedy
we
gets in
checked,
my fucking Twitter replies.
there's like no social sort of like safety net to really help like teenagers with their pregnancies and all of this other I just don't even understand like how this is even a thing that is of any import. If it doesn't affect your future reproductive capability, I consider it a win. Yes, let's
Well,
not have
who knows if it does? It
children
probably
breeding.
doesn't.
Yeah.
It probably doesn't because there's no data. Because guess what? Nobody's done those studies because nobody wanted to put in an IRB application. And for anybody who's had that particular experience, boy, you don't just do that casually. Um,
Yeah.
Put in an application to an IRB to have minors to instruct minors on how to
Minors to
masturbate so that you
masturbate.
can just
Yeah.
measure their sperm counts, like just in general. And
That's so
so, and he
weird.
had all this stuff
What
about
kind of
testosterone levels. I mean, Juliet, it's all
what kind
maha
of
bullshit.
pervert like does this? Like this is such a weird study. Like I there's so many more
There's a
interesting questions to ask rather than this
yeah, but
particular question
that's
that
not
all
what he
it like
posted.
borders on like sexual sort of I don't
It is
know. It
gross.
feels inappropriate. Yeah.
It's gross and it's weird. I'll go further than saying it's inappropriate. It's like, what the fuck? Like, why? It's not scientifically important. It's weird. It's really creepy to be obsessed not just with teen dicks and masturbation, but with like their fertility. I mean, I was talking to Alexi about my husband about this when this happened. And so I have two stepsons. They're both adults. My husband's older than me. And I like literally have never once thought about their sperm counts until this thing came out. And the only reason I did think about their sperm counts was because I was like, have I ever thought about their sperm counts? Like, absolutely not. And so then I asked Alexi, I'm like, have you ever worried about like the kids' sperm counts? And he was like, what? Like, why? What are you talking about? Like, as far as I'm concerned, also, both of my kids are adults. Like, if they have issues with fertility that they need to talk to us about, they have the choice to come talk to us about them. Like,
And they
I just
also
don't
have the choice to
understand why
keep
this
it
is
to
like
be
that
have it be their own
secretary.
business as well,
The
right? Like
Secretary of Health and Human Services is implying that this, like, asking this really weird, fucking unimportant, invasive question is like a good use of US taxpayer money. And all of our, you know, now increasingly unemployed scientific brain power being devoted to like dumbass questions. So, you know what, Juliet, though, I'd love to keep chatting with you about sperm, but we've got 15 minutes left before
oh my god,
people
and we have not talked
jump
about
off.
measles at all.
We
Oh my
barely
god.
talked about measles. And so I'm, uh, I'm not going anywhere tonight. I'm going to be up all night because I've got students who are finishing their theses, and I remember what that experience was like, so I'm gonna be there for them. So,
I mean, I guess
more
one
measles
thing
Manhattans in my future, maybe.
yeah, I
Maybe
think
some
one
coffee
thing that
too.
I taught on measles on Monday with my course like host responses to viral pathogens. And I guess there may be two things I might want to that I will tell the audience. And it's that at one point, we did not have measles in the United States of America. That was the year 2000. It was deemed that measles had been eliminated. And what elimination means is that like you've gone a year without any measles popping up. So like
Well,
we were
no,
on
actually,
the
without endemic transmission. I mean,
right, right,
measles
and
can
then
pop
yeah,
up imported, but it can't
but
spread.
yeah, exactly. And so we had that, and now we are at the point where people are not vaccinating at all. I mean, and people are so fearful because of all this misinformation, and now the cases are rising again. I mean, there was a point where measles was killing a lot of kids. Like we talked 2 weeks ago about polio, where there was a point where everyone was so scared of polio, and now people are like, "What is polio? Is that even real?" It's like,
It's
yeah,
good
it's
for you.
real, and it can come back. And then measles is like the most the most infectious virus that we've ever characterized as a culture. I'm sure there are other viruses that are more, but we just don't know of them. But measles, if you enter a room and you have measles, um, about, I mean, 8 out of 9 people are going to get measles from you if they're not vaccinated. Like, that's huge. Like, it's called R naught. Um, so yeah,
12 to
those are the two things.
18, according to some studies.
Yes, that's true too. Yeah. So it varies, and I'm sure it varies on population because like a lot of the ways that you know acquiring an infection occurs will have to do with your health, your genetics, all of this other stuff like what you've been exposed to in the past. But yeah, yeah, measles. I am I am not feeling good about this at all. Like it's gonna get so bad.
No, I
Um,
mean, it's really terrible, Juliet. Like, right now in South Carolina, I mean, they within 4 months, like, they're almost at 900 cases in South Carolina, and it's 4.5 months, roughly, since the first case showed up there. Um, and I mean, it's just insane because some of the communities in South Carolina have vaccination rates of 20% or lower.
wow, that
These are
is
private
really
schools,
low.
but in some counties where, this is the thing that drives me crazy, right? Like, vaccine mandates are not mandates. They're requirements to access certain services, yes. That's for the good of everybody. That's like if you wanna go on the fucking freeway in your car, like, you put your seatbelt on. If you wanna send your kid to public school, then you get
You
them vaccinated.
vaccinate.
But guess what? You can send them to private school and you don't have to vaccinate. And that's the situation in South Carolina where there are these schools that are private schools that have these incredibly low vaccination rates. But the public schools in that area have done away, and this is basically local politics. This is why local politics, I think, are really also as important as federal politics.
If not more
In
so, to be honest. Yeah.
in Gaithersburg County, South Carolina, that's where most of the cases are happening. Some of the cases were introduced to a religious community that distrusts vaccination for religious reasons. So there are a lot of religious exemptions. But one of the reasons that it's coming out in South Carolina is that for several years, due to, freedom, religious communities and freedom lobbyists, and let's face it, fucking Russians are basically carrying out disinformation campaigns to make it so that vaccines are not something that you can choose, but something that's the government is forcing on you. And so you need to disregard the laws of this government and not vaccinate. And you don't have to be a criminal. You don't have to you don't have to be like the equivalent of me on like the other side or somebody even more like active than me. You don't have to be screaming about it like all the time. But like don't vaccinate your kids. What harm can it do?
Yeah,
Who
yeah,
was the
no,
last person
yeah,
you knew who had measles? Well,
yeah.
nobody. And you know, 5 or 6 years ago when the kids who are getting measles now were born, of course there was no measles. So people who don't know how any of this stuff works will look around and say, "I don't think measles is a threat. I don't think it matters if I vaccinate or not." Like, what about my baby? I don't wanna take these risks. It sounds like there's stuff that's not known about this. And, you know, I've never heard of anybody getting measles. I don't know anything
Right.
about
Yeah,
measles.
because vaccines work, right? And that's the beauty of it. Like they okay, so there's another thing I did want to point out is that there is a side effect of getting measles. It's called SSPE. So oh God, let me look up the name right now. Um,
Oh, it's subacute sclerosing pan something
Pan
encephalitis.
acute something another,
Yeah,
encephalitis.
I just
Yes, but like
it's fucking terrible. Explain
it's
it, Juliet.
absolutely terrible, and you don't
SSPE.
even know. Like you could have gotten measles when you were like a 1-year old, and then like 10 years later or 20 years later, you will just fall into a coma and lose all of your like neurological sensations and everything. But you know, like you start off a day being completely vibrant and then just fall into freaking like yeah, you're completely disabled from the head down, and that's because of measles. And people don't know this because nobody talks about it. And I think the risk is like 1 in 10,000. That's a pretty big risk, right? Like
I
I
think it's
would rather,
100,000, but
yeah.
it's 100,000. Yeah.
Okay, right.
It's not
Okay,
that it's
so
not that common, but
so
the
that's
fact
not,
is,
it's
like,
not that common, but one in a hundred
what's not
thousand
that
based on
common
our
mean?
globe, like come on, like that's really
But
I wouldn't
also,
play that game, and I think most parents, if they knew that this was a potential issue, they would decide differently. It's just that they don't think that there are any consequences to measles, right? They're like, "All right, you're gonna get itchy and you don't
it's
feel good."
natural.
Yeah, but no, like you could set up your child for years later actually succumbing to encephalitis. Just that pops up out of nowhere.
Yeah. I
Uh
mean, it's really horrible. Like, I don't think people understand that these diseases, we made these vaccines and they are still recommended after 80 years, even though we have none of them in this country for a reason. Like, we don't want them coming back. It is a real crisis that they are going to come back. And I keep saying this. I said it to Vincent last night. He wasn't, I don't think he was that impressed with my like little catchphrase. But measles will be first and polio will be worse. And, we talked about polio last time. We'll talk about it again, I'm sure. We'll
mhm.
talk about measles again, I'm sure. Next time I'll do more research on paramyxovirus and we can talk about, like, I don't know, Ben Hurley and Paul Dupre's papers. But yeah, paramyxovirus shit. We can talk about whatever people want to hear about. And today we're just clearly just shooting the shit. So let us know how you feel about all of this because we're more than happy to like also have a more serious discussion. But one of the things we wanted to do was actually have like a discussion between friends and colleagues that would kind of, I think, give people a little insight into what we're thinking about this particular moment and to how we're dealing with this in terms of our expertise. Oh boy, Juliet just her internet connection failed. So I'm just going to talk. Um, we were going to talk about measles. And one of the reasons that Juliet and I decided to have this Substack Live is that Juliet has been, struggling. She's not struggling to keep up with the news, but she's not like me, basically, where she's on top of all of this stuff and she knows exactly like how bad all of this stuff is, but she's not like up to date on like the most recent shit. And so one of the things we talked about was I am desperate to talk to people about science. I am so busy spending every single day looking at the horrors that are being inflicted on my profession, and trying to figure out like what I can do about this and how I can help people understand like how horrible this is. Because most of these horrible things are really like the anti-vax movement saying that things are horrible when they're not. And so one of the things that Juliet and I really wanted to do, and she just sent me two texts saying that her phone got too hot. And I kind of love that because now our Substack Live is just too hot for Juliet to like even finish it. Good God, woman. But I think it's really important for people to understand like how this is affecting us as human beings as well as scientists. So I'm sorry that the whole episode today was not about measles. I don't think Juliet's going to be coming back, and that's okay because I got to go eat dinner and make another measles Manhattan anyways. But I just wanted to thank all of you for coming to this. And next episode, please let us know in the comments to this what you'd like to hear about. Like, I would love to have a more structured, but still casual and fun discussion with Juliet about all of this stuff in scientific terms and continue to make signature cocktails and continue to help all of you get good information. Holy shit, Juliet is back. Yes, Juliet, are you coming back in? We're just gonna say goodbye. You guys are wonderful. Okay, she's gone. We're gone. You guys are wonderful, and, I apologize for being such a poor content creator. To be honest, this is, as we discussed tonight, my hobby. I am a professional academic, so I know how to publish a good journal article and get through peer review. But I'm not so good at Substack Lives. So I promise we'll be better for the next episode. Please let us know what you thought. Please let us know what you'd like to hear about. We are going to structure it more. We're still going to have a lot of fun because the world's a hellscape right now, and we really do need to have fun. And I'm so glad to those of you, I'm so thankful to those of you who showed up tonight. I honestly don't know what I'm doing. I'm the worst social media celebrity of all time. I get told constantly that I'm like a COVID media whore, and I'm actually really terrible at it. And I'm very bad at monetizing it. So, what I really do want to do is communicate things to you guys that's going to be useful for you because we all need to be in this fight. And one last thing, I'm on the steering committee of the Save America movement for public health and science. We are about to start taking off on a lot of activities to help keep you guys informed about all of these things that are affecting us. So I'd really love it if you got into the comments too and told me what would be most useful for you in that regard. I'm already trying to tell people what I think, actions are needed in order to fight back against all of this. But I really love to hear from you guys what we could be doing better because we can't fight this unless we do it as a community. And that means all of us together. So please give us some feedback and we'll see you week after next. Probably back on Mondays again, but we'll see. Love you, Juliet. Dr. Morrison can be found on PubMed, where she's published many excellent papers about macrophages, host responses to a protease of poliovirus, and let's see, dengue, flu. She's brilliant. Go check her out. And you can check me out here at Substack at Rasmussen Retorts, Substack.com, whatever. If you're into Substack, I'm learning. Sorry, guys. I'm really competent at Twitter, and now it's a Nazi hell site, and it's like number 8 on my priority list to cope with in terms of all the things I'm upset about. Good night, you guys. Take care. Get your MMR vaccine if you have not. Also, still not too late to get your flu shot. Good night. Oh, shit. I can't even turn off my phone.